So let's talk about the uterine tube. Now the uterine tube is composed of the infundibulum, which of course bears the fimbriae, these projections, these finger-like projections over the ovary. Exactly next we're going to find the ampulla of the uterine tube, next the isthmus, and lastly the uterine portion of the uterine tube. Now the uterine tube is quite a simple structure. It's also called oviduct. Now it's composed of the mucosa, the outer mucosa, which is highly folded and highly coiled. Exactly beneath we're going to find the muscularis layer, and in the end we're going to find of course the serous layer. Now, of course, the epithelium contains, there are two different types of epithelia on the mucosa. There is the PEG cell and the columnar cell, a simple columnar cell. Of course, beneath is always the lamina propria, but of course, always in between the basal membrane between the mucosa and the lamina propria. So the PEG cell or secretory cell secretes nutrients and most importantly the capacitation factors which activate the sperm, and they're responsible for the nutrition of the ovum or the fertilized egg. Now the other cell that we're going to find is the simple columnar ciliated epithelium. The point of the cilia right here is to move propel the ovum or the zygote towards the uterus. This is not of course the major let's say the major event that helps this propelling motion. It can also be found that in women that do not have the capacity to have the cilia, the amotile cilia syndrome, they can also move the zygote properly towards the uterus, and this is because of the major musculatory components of the uterine wall, or the uterine tube in fact. So if we actually take a look at the macroscopy, we're going to see exactly the mucosa. Example, this is a highly folded and coiled structure. So if we zoom in, we're going to find exactly the feature that we saw before. So by zooming in, we actually see two different let's say population of cells, the ciliated and non-ciliated. It's not always somehow easy to distinguish between the two because of the high amount of overlapping between one and the other. And the easiest way for you to do that is to find a very clean section, very thin section where you can see just only one, maximally one layer. For example, this one, still not the best, but you can see some cilia present on the top with interspersed spaces that do not have it. So we can assume that the ones that do not have it are the PEG cells, the secretory cells. And the other one, the simple columnar cells, as always. Exactly beneath is always the lamina propria. And on the muscularis, we're going to find two different layers. Of course, the first one is going to be the inner circular layer, which is approximately right here. And then we're going to find the more outer, which is going to be more spiral between, like there's going to be, let's say, a complexity between the inner circular and somehow spirally formed layer or more oblique layer, and the other one is going to be the more longitudinal section. Of course, it's not that clear because they're overlapping a lot, but the difference is of course because of the circular is going to have more cross sections, more the appearance of a cross section of smooth muscle, and the long is going to be a more clear section of the smooth muscles. And last we're going to find the serosa, of course, which is typically this, of course, the loose connective tissue along with the vessels. And in the last layer we're going to find the mesothelial cells, of course, covering up the whole outer layer, the whole outer surface of the oviduct. So let's move on. Next up, we're going to talk about the fertilization events. Now, the first event that's going to happen is after the sperm, of course, the ejaculation, the sperm attachment on the zona pellucida. In this case, we're going to have the acrosomal activation. Now, the acrosomal activation, the process is going to be the release of hydrolytic enzymes that penetrate the zona pellucida and eventually reach the layer of the perivitelline space. This is nothing more than just the space between the zona pellucida and the specific protein receptors that surround the plasma membrane of the oocyte. The point here is that we're going to have the penetration of the nucleus, the sperm nucleus, after the attachment after the penetration, of course, of the sperm to the cell membrane of the oocyte. One interesting feature is that we have these cortical granules. These cortical granules are in fact products of the oocytes that, as you can see in the picture, also are always located nearby the plasma membrane, and they are actually exocytosed the moment we have the penetration of the sperm nucleus to the oocyte. And this, of course, prevents the polyspermy, prevents more sperms to go and, of course, and to invade with their nucleus the egg cytoplasm and in the end the oocyte. So these are the basic features of the fertilization process, and this of course usually takes place in the uterine tube. Now next up we're going to talk about the uterus. Now the uterus is a structure that has 3 to 4 layers. Why 3 to 4? Because the mucosa typically tends to have tends to be subdivided into two layers, the basal and the functional. So we're going to have, as always, the mucosal layer, and we're going to exactly have the myometrium beneath. And of course, is the endometrium, myometrium, and perimetrium, the outermost layer. Of course, when we talk about the terms functional and basal layer of the endometrium, this has to do with the of course, with the mitotic rate of regeneration of the functional layer, and the functional layer itself is in fact the one that undergoes continual changes throughout the whole menstrual cycle. So the basic features we're going to see in both the layers is going to be the uterine glands. Now the uterine glands are of course covered by ciliated and non-ciliated cells, primarily ciliated columnar cells, single columnar cells. And the other one is going to be, of course, the spiral artery. Now, the straight arteries and spiral arteries, there is a difference between the two. The spiral artery tends to penetrate the functional layer and tends to perfuse the thickened endometrium we're going to see throughout the whole menstrual level. So it is important to really understand that the functional layer is a highly adaptive layer that changes completely when it comes to thickness and in layers, in the thickness of the layer. Now these are the typical again features. The endometrium is right here, myometrium and perimetrium on the outermost layer. What is interesting to notice is of course the mucosa. Now the mucosa, I told you before, is a typically ciliated cells with with sparse non-ciliated cells. The uterine gland penetrates from the basal layer to the very top of the functional layer, and the continuation of course the mucosa is again the similar mucosa, exactly the same. Now, what is of course, this is the thickness of the myometrium. The myometrium and the functional layer undergo continual changes. The difference is that based on the menstrual level, the menstrual cycle, the point of the menstrual cycle, we're going to actually review these changes in just a bit. So there are three phases of the menstrual cycle. The proliferative, in which of course the cells of the endometrium tend to mitotically divide continuously, of the basal layer tend to mitotically divide and to start to thicken the layer of the endometrium. Typically this is going to start the thinnest portion is going to be 0.5 millimeters, and in the end we're going to reach up to 5 millimeters. So this is a tenfold increase. So it's a quite big proliferative tendency. Now the next layer is going to be the secretory. This is the difference. Exactly, this is the same magnification in both pictures. So we can see the difference between the sizes of the two. This is tenfold, statistically more or less tenfold increase of the thickness. And during the secretory phase, we're going to find some specific features that are being affected. Specifically, we're going to have the production of progesterone, which will stimulate the epithelial cells and start to accumulate glycogen. Glycogen, of course, is always, you should think about it as an energy source. It's the cells that do not receive increased amount of perfusion and increased amount of oxygenation nutrients. And as a consequence, because of this highly accumulative nature, you're going to start becoming coiled glands. The uterine glands that were straight now start to become more coiled because of this aggregation of glycogen. Now, the point of why do we care so much about the uterine secretion, the uterine glands? Well, because these uterine secretions are in fact the major source of nutrition for the embryo. Importantly speaking, progesterone also inhibits the contractions of the myometrium. The point is that we want to prevent any damage to any sort of implication to the implantation of the embryo that will of course take place in the endometrium. And last, we're going to find the menstrual phase. This is of course going to take place in the condition that we do not have the fertilization and the implantation of the embryo on the endometrium. And in this case, we're going to have the lower amount of progesterone. Of course, this is going to have as a consequence the destruction of the increased muscle spasm contractions, and these are of course the typical clinical features seen as contraction pains in the menstrual cycle. And the second most important is going to be the increased production of prostaglandins by the arterial cells, and as a consequence, because of these prostaglandins, we're going to have vasoconstriction and local hypoxia. The result of local hypoxia and vasoconstriction practically is going to be the ischemic death of the endometrium, and we're going to have the shedding off of very, very big portions of the endometrium. So the point of the whole cycle is of course started by the estrogen levels going up and up and up and peaking, along with, of course, with this peak of the estrogen is going to have the peak of the LH that will induce the ovulation. After the ovulation takes place, we're going to have the formation of the corpus luteum. By that moment, we're going to have the maintenance and the production of the progesterone from the corpus luteum. The difference is that this corpus luteum will not tend to produce progesterone continuously until the end. So if the ovulation takes place, if the fertilization takes place, the embryo is going to in fact produce the human chorionic gonadotropin hormone, which of course will start to induce and maintain the progesterone levels, and will continue inducing the production of progesterone from the corpus luteum and maintain the progesterone at a high enough level in order to prevent, of course, the normal menstrual cycle to complete. The typical menstrual cycle is approximately 28 days, and approximately in the 14th day we're going to have ovulation. So this is the difference between the hormonal changes. As you can see by the picture, of course, right here, this graph, again taken from John Quera, we're going to see that the functional layer is the one that changes thickness from 0.5 to 5 millimeters. This is a very big difference, and this is the one that's going to be pretty much affected by this changes of the menstrual cycle. So let's visualize uterus in the macroscope. Now this is in this case we're going to see the secretory phase, where we're going to have the thickest layer of the endometrium in this case. Now this is the endometrium, this is the myometrium, and the last point we should see the perimetrium. In this case I don't see that it's actually that close and that visible. In this case we'll have a serosa, so the other part is going to be, the most parts of the uterus is going to be serosa, and some areas are going to have tunica adventitia. Now the myometrium is going to be composed of 3 different layers. The first, the longitudinal layer, then the more circular or spiral one, and lastly again the longitudinal section. Now if we zoom in, we're not going to see exactly the typical feature. We're going to see more complex structure, more overlapping structure with different features of different orientations. For example, this is the typical longitudinal section. This is a more circular, more, let's say, oblique manner. But typically speaking, we should be able to visualize in a clean section, in a very, very well oriented section, we should be able to see the distinct layers of all 3. More typically speaking, we should find that the outermost layer has the most, let's say, correct orientation, the more typical orientation of the longitudinal section, and this is what you should actually visualize, typically speaking. Now let's see if we can see in this section the serosa of the perimetrium. Not that clear and not that easily visible. So let's keep in mind that we should find the serosa in most areas of the uterus, and in others we're going to find the interstitial. Now the most important part we should discuss is the changes that the uterus undergoes throughout the whole menstruation. We discussed already about the endometrium, that it thickens continuously by the function of progesterone. The other difference, the other change that happens during pregnancy in this case would be in the myometrium. The myometrium, in fact, during pregnancy is going to start to undergo cellular hyperplasia, which is the mitotic rate, the increased mitotic rate of the smooth muscles. We're going to have increased production of collagen, and these 2 phenomena are going to actually result into the thickening, into the hardening of the uterine wall. Of course, this is necessary to allow for parturition, for practically giving birth. So we can see that the endometrium is going to be composed of, again, of the thick functional layer and the very smaller and proportionally smaller basal layer. Now the basal layer doesn't undergo changes like that much as in comparison to the functional. This is going to be, again, the area of the mitotic rate regeneration. And on top we're going to start seeing, of course, these highly coiled structures of the uterine glands, and on top we're going to find the ciliegranal epithelium on the top. Now in this case we're going to start seeing, again, the columnar epithelia. Exactly, we're going to find the lamina propria with interspersed, mostly, typically speaking, the typical features, the stellate cells, macrophages, lymphocytes, and in some occasions we're going to find some fibroblasts in this case. But the most important feature to remember is that this functional layer is going to undergo continuous change for the menstrual cycle, and the important feature is that these cells, of course, start to accumulate glycogen, and as a consequence receive this more coiled appearance of the endometrium in the uterus. So let's move on. Now next up we're going to talk about the vagina. The vagina is an organ that is being continuously bathed in liquids, and the point is that because we have this liquid nature, this more wet area, we're going to tend to have the non-keratinized epithelium, stratified squamous non-keratinized epithelium. Always remember this, of course, this let's say a train of thought. Whenever you have a wet area, you should directly think that you should have non-keratinized epithelium. For example, esophagus, upper part of the esophagus is going to be, of course, the stratified squamous non-keratinized. In areas where we're going to have a low amount of liquids, and of course we're going to tend to have the loss, the threat of losing liquids and losing water, this is going to be, of course, a typical location in the skin. We're going to have a keratinized structure. This is how you should remember, you should easily identify this area. because let's be honest, the vagina itself is not going to have some unique physiological feature to help you to understand and actually identify it. So the typical feature would be, of course, to think that you're going to have this non-keratinized structure, the typical appearance of it we've already discussed in multiple videos. And of course, this is going to be again the typical feature with the presence of nuclei on the top, on the more outermost layer of the specific structure. So let's visualize the rest of the vagina in the microscope, shall we? So, as we said before, we're going to have again the typical stratified squamous non-keratinized epithelium, which is going to be very easily discernible by the presence of nuclei on the very top layer of the stratified epithelium. Again, we can actually visualize it very, very easily. Now, as always, we're going to have beneath it the basal membrane and lamina propria. Basal membrane is not, of course, visible in light microscopy, but still this is a tip feature you should mention always. Again, we're going to have the lamina propria beneath it, the loose connective tissue, along with fibroblasts and other elements. Exactly beneath, we're going to start seeing the muscularis layer. Of course, this is the inner circular layer of the muscularis, and exactly beneath we should find the more, let's say, complex and the more typically seen, of course, the longitudinal sections of the smooth muscle cells. Now, practically speaking, it's not that always this typical features of the layers. You're going to see overlaps. You're going to see things that are not typically or not actually textbook pictures, but this is real histology, and this is what you should be actually accustomed to. And typically speaking, the outermost layer is going to be the adventitia. In this section, it's not that easy to visualize them, but we can see actually the vessels right here, the very typical morphology of the vessel right here. And again, this should be the location of the adventitia, which should have the loose connective tissue along with the, of course, some vessels and some adipocytes. So let's move on. So let's talk about the mammary glands. Now, the mammary glands, of course, have different forms when it comes to the, of course, activity. Now, they can be mature but are inactive. Of course, this is going to be reflected in the size of the lobules. We're going to see exactly what we mean by lobules. By early pregnancy, we're going to start seeing the, let's say, the enlargement of the alveoli, and it keeps on. This enlargement keeps on until the very, very point of parturition and the lactating procedure that initiates the lactating procedure. We're going to have a progressive let's say, enlargement of the alveolar ducts and, of course, the alveoli themselves, the acini themselves. Acini. And lastly, we're going to see the regression after weaning. This is going to be, we're going to see the apoptotic points here and apoptotic signals and features. And we're going to see the destruction and self-destruction of this, of the glands. Now, the basic, let's say, logic, of course, is simply as a gland. Again, this works exactly the same like a gland. We're going to have again the secretory portion, the acinus, and the duct. As always, the acinus is the one that produces actively the content, the components of the specific, let's say, product. In this case, we're going to have high amounts of lactose. We're going to find high amounts of proteins, and of course, we're going to find high amount of lipid-rich contents. So the protein components are going to be produced by the merocrine secretion, which practically means the degranulation of these proteins in the lumen. The lipid products, the lipid components are going to be secreted by the apocrine, which means there's going to be a small sloughing off, a small part of the cell membrane being lost in the procedure, and then we're going to find the typical appearance of the locular tall cuboidal cells. Then last, we're going to find the ducts, the conducting portion, which enriches and of course deposits other components. Specifically, for example, here we're going to see that in between the ducts we're going to have, and in between the acini, we're going to find, of course, plasma cells. Of course, these are the differentiated, specialized B lymphocytes that produce antibodies. Specifically, in this case, we're going to have the typical mucosa-associated antibodies, the IgAs, and this is going to be the way to ensure the passive immune reaction from the mother to the infant. Now, we're going to find, of course, as typical as always in the glands, the two features: the stroma, which is the part of the organism, the part of the organ that supports the structure, and the parenchyma, which is, of course, the functionally active part. Now, the functionally active part in this case is going to be the alveolus, the alveoli. Now, in these cases, we're going to have these alveoli are going to be the terminal, also coincide with the terminal ductules. Now, all of these terminal ducts are going to practically converge to form the intralobular ducts. As the name suggests, intralobular, we can actually identify and understand that this is a structure that is lobulated, and in fact, all these lobulated structures are going to have the intralobular ducts, the ducts that converge from the terminal ductules And in the end, after these intralobular ducts converge, they will form the lactiferous ducts, the bigger ducts that connect more than one lobules. And the last, all of the milk that is being produced by the mammary gland will be aggregated and controlled and practically stored partly in the lactiferous sinus. The sinus, of course, means that there is an empty space, and space specifically that allows the deposition of contents within it. So this is the typical feature of the mammary gland. This can be easily confused with the thyroid because we're going to have similar features, similar colors, but there are, of course, other components in the thyroid that differentiate the mammary gland with the thyroid. And in this case, we should be able to visualize that this is a very clear and lobular duct. We can see the trabecula, the presence of connective tissue in between the specific ductules, and of course the alveoli in this case. And the difference is that we're going to see inside the volumetric difference that we see before pregnancy and after pregnancy and during pregnancy is actually visible in the even in the macroscopic level. The lactating one is this one on the left, and the right one is the non-lactating. As you can see, the difference in the volume is massive. The difference, of course, is that in the presence of the hormonal actions of the prolactin, of the progesterone, of the estrogen, we're going to have the enlargement of the alveoli, specifically in the terminal ductules. So we're going to have a higher and more productive mammary gland. In fact, it's going to be active to produce all of these nutrients that should be that are necessary, of course, for the infant to survive. So here we have the non-lactating one, which is in fact filled with dense connective tissue and very, very few signs of ducts. In fact, these ducts are going to be the lactiferous ducts, the biggest ducts in the mammary gland. And this is a typical feature of the post-lactational apoptosis. This is exactly what the cell undergoes, the apoptosis, the death of the cell. After, of course, we're going to have the regression of the hormones that, of course, maintained this whole hormonal level and this balance that actually maintained the size and the volume of the active lactating glands. So let's visualize the structure in the macroscope. So this is again the non-lactating, and this is the lactating. So let's zoom in the lactating. So if we go exactly and zoom in, we're going to see that the lobulated morphology of the ducts and of the whole mammary gland. And if we zoom in, we can actually start to see the alveoli, specifically the acinar parts, are also visible and can be seen in the sections. So here we're going to see the typical alveolar structure of the mammary gland. We're going to find cuboidal, two locoluminar cells in this case, and in the center we're going to find this eosinophilic material. This is, of course, the milk that is being produced by the mammary gland. And interspersed between them, we're going to find smooth muscles. We're going to find also, very importantly, myoepithelial cells that in fact will help and assist the movement of milk from the acinus towards the ductular portion, the conductory portion of the glands. And in between them, we should see bigger sections, bigger structures that are in fact that constitute the ducts, the intralobular ducts. Let me try to find one. So this is one right here. We can actually distinguish the basic difference between the typical the alveolus with the duct, with of course the intralobular duct, by the amount and the layers of cells that we have, cuboidal to locoluminar, of course, in the same case. And if we see stratified, this is exactly the first indication, the first giveaway that this is in fact the terminal ductile and not the intralobular duct and not just the terminal ductile. So it's not that easy in this cases to identify them both, to identify a clear terminal ductile with intralobular ductile, because of two reasons: because of the overlapping and the overlapping, I mean, of the cells. Sometimes the overlapping can seem that we have the stratified even though it's not. And the second giveaway is that the second problem is going to be of the orientation of the sections. So always try to find the most clear section, the thinnest portion of the section, and be more certain that this is in fact the intralobular duct. Now the next one we should find is the interlobular duct, the lactiferous duct, in fact, that converges. That this is the location where of course the intralobular ducts converge and start to form the bigger ducts. So let's try to identify and to find the lactiferous duct. Now this should be much easier. Why? Because the size of the lactiferous duct is much bigger, and in fact can be visible even by low magnification. For example, this is one right here. You can see that in comparison diameter of one lactiferous duct, and of course the smaller alveoli right here and the small depositions of the milk in the lumen. So if we zoom in, we can actually find a very unique epithelium. Again, the double layer of cuboidal to columnar epithelia. And in this case, we're going to find that's more thin section and more, of course, easier to distinguish, that the pure certification is actually visible. For example, this one This is a typical morphology of the cuboidal, the stratified cuboidal epithelium that is in fact the lactiferous duct. It is surrounded by myoid cells, and exactly surrounding that we're going to find the smooth muscle cells that in fact play a very big role into the contraction and allow for the milk to be excreted through the lactiferous sinus and the areola. So let's visualize, of course, as a last point, the inactive resting mammary gland. And we can see that this actually is filled with dense connective tissue. If we zoom in, we're going to see, of course, big areas of, this is exactly one that we can see, the eosinophilia, the high amount of collagen fibers, and of course interspersed vessels. Interspersed, this is a ductal, sorry, this is not a vessel, this is a ductal. And generally speaking, we should find some vessels, and here we're going to find in fact much clearer sections of the ducts, of the lactiferous ducts, because they're more clear in this section. We're going to find the more typical appearance of the stratified cuboidal to columnar epithelium in this case, and this is a much more compressed morphology because of the amount of the smooth muscles because of the amount of the connective tissue surrounding it. Thank you very much.